Welcome, everyone, to Introduction to ALS MND Genetics. My name is Kathy Cummings, and I'm the Executive Director of the International Alliance of ALS MND Associations. We are very privileged today to have some fantastic speakers to give us the information we need to know in our community about genetics and the role they play in ALS and MND. Jess, could you move the slide forward one, please? I would like to thank our sponsor, Biogen. Without Biogen's support, we could not have done the work we've done in terms of delivering genetics webinars. We do have a series of webinars coming up, and Christiana Salmon, our moderator, will talk to you a little more about the other webinars that are coming in this stream. And without further ado, I will turn the webinar over to our fantastic moderators, Bruce Virgo and Christiana Salmon.
Thank you, Kathy. So my name is Christiana Salmon, and I'm the National Programs Manager for Genetic ALS at the Montreal Neurological Hospital, and I'm leading a Canadian initiative to improve access to genetic testing for people living with ALS. And so I'm pleased to be hosting this series of webinars for the Alliance, as it's a topic I'm clearly extremely passionate about. We're seeing a boom of therapies being developed for ALS right now, and a number of those are specifically targeting the genetic forms of the disease. It's becoming increasingly clear that there's an underlying genetics role that genetics plays in all of ALS. And so the more that we can understand about the genetic forms of the disease and how to effectively treat them, the better we can understand the disease spectrum as a whole. We also recognize that this information or access to specialists is not available globally. And so we hope that this series of webinars will start to fill that gap. I have my wonderful co-moderator here with me today, Mr. Bruce Virgo. Bruce is a lawyer by training and an active advocate for MND awareness and research. He is also on the PALS-CALS Advisory Council for the International Alliance of ALS MND Associations. As a person living with MND, Bruce would like to take a moment to share his perspective on genetic testing.
Thank you, Christiana. I'm honored to be asked to co-moderate today's webinar. As Christiana said, I've been involved with awareness and research since I was diagnosed in 2014. I signed up immediately to the MND register here in Scotland in order to participate in MND specific research. The same year I was invited to join a genetic research project conducted by the Ewan MacDonald Research Foundation. The foundation is linked to the Anne Rowling neurology clinic that I attend here in Edinburgh. That's a charitable clinical research facility of the Edinburgh University. As that project was totally anonymized, my actual results were not made available to me. And so the information on genetic testing was restricted to printed material, and no counseling was required. I followed the progress with genetic research into MND ALS ever since. I am aware from a previous unrelated health issue in Australia of the impact genetic testing can have when results are revealed and the potential implication for others. And I had undergone pre-test genetic counseling at that time. I had an opportunity in 2018 for a further genetic assessment related to my MND, which was offered by the Anne Rowland Clinic. I'm very interested and keen to hear both our speakers as they expand on an introduction to genes and genetic testing and development in our disease. Although I was counseled in 2018, I was still very keen to proceed not only for my own personal reasons, but also to aid in further genetic understanding of my form of MND. Thank you, Christiana.
Thank you, Bruce. Now, before we introduce our wonderful speakers today, we'd like to provide a little bit of background on how this webinar series came to be. The Alliance hosted a roundtable discussion on genetics and ALS MND in March of this year. This roundtable brought together experts in the field, people living with ALS MND, as well as industry partners. As a result of that roundtable, we had several outcomes. I've listed the outcomes here, but in summary, it was to create resources in multiple languages on genetics and ALS MND, develop education programs about genetics, which is the webinar series that we're starting today, communicate the statement of rights of people living with ALS MND, which was updated to include a statement on genetics, which I'll share momentarily, and to create clinical guidelines for genetic counseling and testing, which a group of clinicians is taking on. The fundamental rights of people with ALS MND and caregivers has been updated to say, people living with ALS MND have the right to have access upon their diagnosis to up-to-date education about clinical genetics and ALS MND, genetic counseling, genetic testing, safeguards against genetic discrimination, and subject to education and counseling, blood relatives should also be given that same access where relevant. So this series of webinars was created to start to address that gap in terms of information out there for the community of people living with ALS MND and their family on ALS genetics. Today, we're having the Introduction to ALS MND Genetics webinar. In 2 weeks' time, we encourage you to register for the Genetic Counseling and Testing webinar. And finally, in December, we'll have a webinar on ethical considerations for genetic testing in ALS MND. Today's learning objectives are to understand basic concepts in genetics, such as what is DNA? How do we inherit our genes? What is a genetic mutation? We'd also like to understand where we are with our understanding of ALS MND genetics today. How do we discover these genes that are associated with the disease? What are those genes? We'll provide an overview of more complex concepts in ALS MND genetics and highlight some strategies for genetically targeted therapies. And with that, I'd like to introduce our speakers. Dr. Kelly Williams is a senior research fellow and the group leader of the Genomics and Bioinformatics research team within the Multidisciplinary Care Center for MND Research at Macquarie University in Sydney, Australia. For the past 15 years, she has focused on determining the genetic basis of ALS MND, and her work has contributed to seminal discoveries and identification of causal mutations in ALS-associated genes. Dr. Williams's research has translated into improving clinical practice with these disease genes, now added to diagnostic tests worldwide, including pre-implantation genetic diagnosis and pre-symptomatic testing. And Bruce, would you like to introduce Dr. Al-Shalabi?
Certainly, thank you, Christiana. Professor Amar Al-Chalabi probably doesn't need that much introduction to the wider ALS MND community, given he's a tireless researcher specializing in ALS and MND, and has presented globally on many occasions with respect to our disease. Amar is a consultant neurologist at King's College Hospital in London, as well as a senior investigator and professor of neurology and complex disease genetics at the Morrisville Clinical Neuroscience Institute at King's College. In addition, he's a director of the King's Motor Neuron Disease Care and Research Center. His research team works on finding the causes of ALS, potential treatments, and what might influence the way the disease manifests and progresses, particularly where this involves genes, environmental exposure, or lifestyle. He runs a large clinical research program along with clinical trials, the details of which are set out in his biography you will have seen on registering for this webinar. Also set out in that bio are the many international awards he has received and honorary positions he has held relating to his work into our disease.
And one final housekeeping note before I turn the slide deck over to Kelly. Regarding comments and questions, you have a few options. You can put them in the Zoom chat. There's also a Q&A panel. And if you're watching us from the Facebook live stream, you can put your questions or comments in there. These will all be passed on to us, the moderators, to bring up to the speakers at the end. And with that, I will turn it over to Kelly.
Okay, thank you. I'm going to share my screen. Okay, Christiana, I just want to check that's a correct slide.
Yep.
Fantastic, fantastic. All right, thank you very much. So I've been studying and researching human genetics for the last almost 20 years, so since I finished high school. But really to be able to grasp the key elements of genetics, we really only need to know what genes are, where they're located, and how they're actually inherited. So the best place to start is with our DNA. So DNA, it's the blueprint of life. It contains all the information that we need to make us, us. And it exists in every single cell in our body. So in our hair cells, our skin cells, and blood cells, we have an identical copy of our entire human genome in every single cell. So our genome, it's made up of around 3 billion units of DNA code, and they're all lined up next to each other. And there are only actually four different units that make up our DNA. So these, so as geneticists, we simplify these to A, T, C, and G, and only those 4 units make up all 3 billion units of our code. So if we imagine that our DNA code is like a ribbon, in order to fit it into every cell, because it's 3 billion DNA units long, we need to be able to wrap it really tightly. So the ribbon, it's wrapped quite tightly into what we call chromosomes. And you can see a figure of the chromosome here. And so humans have 23 pairs of chromosomes, so 46 in total. So these 23 pairs, one set we inherit from our mother and one set we inherit from our father. And so these 46 chromosomes exist in every single cell in our body. So to give you an analogy of our genome, if we were to actually start typing out our entire genetic code, we would end up getting about 6,000 units of DNA on an A4 page. But to cover our entire human genome, we'd need 1,500 page books. So we could condense those into chromosomes, but every chromosome would have on average around 40 volumes of these 500 page books of our DNA code. So it is really massive. Now, within our entire genetic code, we have genes. And really, all they are is a certain section, is just a small section of our entire genome. And genes actually only make up around 1 to 2% of the entire human genome. But they are so crucially important because the gene itself contains information for human traits. So things like hair color or eye color or height, the information to make these traits is in our genes. So a gene itself is the basic unit of inheritance. So biologically, what do genes actually do? And they're so important because they provide the information to make the building blocks of our cells, so the biological building blocks of our cells. So here to I guess use the book analogy of our genetic code. If we have a gene, that has the code to make a building block, we don't want to have to read the whole entire book to be able to find out how to make this building block. So we have our blueprint, which is our DNA, and then we have the next stage, which is an intermediary stage called DNA, and that's like a photocopy of the gene. So in our book, a gene may be half a page, 1 page, 10 pages, but we take a photocopy of that, which is our RNA, and we can then give that photocopy, so that information to the cell to make our building blocks. Now, in biology, we call these building blocks proteins, but that's not in the classic sense of when you think of proteins in food. So to give, I guess, a real life example, we have a gene, so we have around 20 to 25,000 genes in our genome. And one of them is called ABO. And this gene has the information, so it encodes the information for our blood type. So ABO is our DNA, and it makes a protein which is called an antigen, a specific antigen that goes on our blood cells. So this ABO gene encodes the information to make a protein, which is an antigen, which sits on the outside of our blood cells, which gives us our blood type. So do we all have the same genes? And you may have heard, in passing conversation, oh, I have the gene for such and such disease. And that's not entirely true, because we don't have different genes. We all have the same genes. We all have the same genes, but we have different versions of the genes. So all of us, every single person here in this webinar, we all have the same genes, but we have different versions of those genes. And in genetics, we call those versions alleles. So I'm going to come back again to the ABO gene, which encodes our blood type. And we have 3 versions. So we have, the first version is version A, or allele A. So this gene here, so this is representative of a gene. This encodes the antigen protein A. So this antigen is what, if we have this blue antigen that's on the outside of our, on our blood cells, we have blood type A, and that's come from allele A. The second version of the ABO gene is slightly different to the first one, and it's called allele B. So this is version B of the ABO gene. It's slightly different, and it encodes a different antigen. And when this antigen is around the outside of your blood cells, you have blood type B. And there's then a third version. So this is slightly different again. It's still the ABO gene, slightly different. This one actually doesn't make an antigen. It doesn't generate a protein. So people that have blood type O, their red blood cells actually do not have antigens around them. So these are 3 different genes making 3 different products or proteins. So why are they different? And it's called genetic variation. So all genetic variation is a small differences in our DNA code. And we have around 4 million of these genetic changes in our DNA code. And it's actually what makes us unique, what makes us diverse and different. However, if one of these genetic changes ends up leading to a detrimental outcome, such as a disease, we would consider this genetic variation or this genetic change a mutation. So this is actually what genetic mutations look like. So again, we're coming back to our ABO gene. If this is it's a snippet of the code, of the genetic code that encodes for the A antigen, and we compare it to the B antigen. So the first few DNA units are the same, and then we get to one that's a different one. And then we have a few more that are same, and then we get to two that are different. And so these small differences end up making a completely different protein. And these are called single nucleotide variants or single nucleotide mutations. So they're happening at a single DNA base, and another word for a DNA unit is a nucleotide. And this type of genetic variation is what we most commonly see in MND and ALS genetics. So then if we look at the third version of our ABO gene, which is the O version, there's actually a section missing. So these 4 DNA units are actually missing in the O version, and this is called a deletion. And in this case here, it ends up making no protein. So this is another type of genetic variant or a genetic mutation. Now, there's a slightly more complex one that is important to explain in the context of MND ALS, and this is called a repeat. So in this hypothetical gene here, we have this section which is a repetitive sequence of TA DNA units, and the hypothetical protein that it makes, that little pink bit, is representative of this repeat section of the gene. So what can happen is this repeat section, they're highly susceptible to mutating. So it's expanded. So rather than having 4 repeats, we now have 10, and we have a different protein. And this type of mutation, of repeat expansion, is actually the most common genetic cause of MND ALS, which I will get to a bit later. So how are genes actually inherited? So we have 2 copies of every gene, because we inherit 1 set of chromosomes from our mother and 1 set from our father. So everyone has 2 copies. So what I've tried to show here, say the male, these are 2 chromosomes, and the colored section is a gene at a specific location on this chromosome. And so it's the same gene, but this male has a yellow version of this gene and a blue version of this gene. And the female, she has 2 versions. She has 2 copies of the pink version of this gene. Now their son, he will inherit one of these versions of this gene from his mother, and one of the versions of this gene from his father. Now, that's as simple as inheritance gets, really. The mode of inheritance, the traits that we have, so their genes can be responsible for our traits, but whether we actually show that trait depends on the mode of inheritance of the gene and of the version. So if a version or a gene is inherited in a dominant fashion, it means we only need 1 copy. So we only have needed to get it from mum or from dad to actually show that trait. Whereas if it's recessive, we need 2 copies. So we need to have inherited it from both parents to have the same version to actually show the trait. We further break this down, and I'm going to go into this in more detail. And there's different types of dominant and recessive based on where the gene is located, what chromosome it's on. So we have sex chromosomes. So of our 23 pairs, 1 pair are our sex chromosomes, which determine whether we're male or female. So if we're female, we have 2 X chromosomes. If we're male, we have an X and a Y. So if we are, the 2 last examples, they're X-linked dominant inheritance and X-linked recessive inheritance. They are both related to our X chromosome, whereas all of our other chromosomes, so all other 22, they're called autosomes. So the first example that I'm going to go through is if genes or a gene, say a gene mutation, so you have a gene that has a version with a detrimental mutation in it. If it's on one of your autosomes, so all of your chromosomes except the sex chromosomes, this is how we see inheritance. So if it's dominant, you only need 1 copy, so one one chromosome that has the mutated version of that gene. And here I've tried to display that by using red. So these chromosomes, this is the same gene across these two individuals. The male has the black version, which is like the normal version of the gene as such. And the female, she has a red version, which is the disease version of the gene, and then the normal version. So she is affected with disease because she has one copy, because this is dominant. So then the children will inherit one of the versions from their father, and they will inherit one of the versions from their mother. And so as a reminder, and I'm going to harp on this a bit, dominant inheritance, one copy will lead to the trait. So in this example here, 50% of the offspring inherit, 50% of the offspring on average will inherit the mutant version of the gene. So autosomal recessive is different because we need two copies. We need a copy inherited from each parent. So you can see here, the mother and the father, they have this red version, so this mutant version of this gene, but they are not affected. They are carriers of this because they need two copies. If we look at their children, there is one child who has inherited it both from mother and the father, and they are the child that is affected. And then we have two children, statistically on average, that would be carriers. Then we're going to go and have a quick look at the X chromosome. So we have sex differences in the X chromosome. So because males only have one. So if it's dominant, we only need one copy. We only need one copy to be affected. If the father is affected in an X-linked disease, again, 50% of the offspring are affected, but in this case, it's only females because males receive their Y chromosome, so they will never receive the X chromosome from their father that has the genetic defect. So in this case, only females will be affected if their father is affected, and the males are completely fine. So X-linked recessive, a really good example for this is color blindness. So color blindness is quite common in population. So it's about 1 in 10 males in European populations are colorblind, and it's recessive and it's carried on the X chromosome. So the gene is on the X chromosome. So if it's mutated, and you're a male, you will be colorblind because you only have one X. So in this case, we have female carriers, males affected. Even though recessive, as I'd said before, you need two copies, it's different with X-linked disease. If the father is affected, we end up having no children affected, but females are carriers. Now, I just want to, so the two families on the side, they're the ones that I've just talked about. But what can happen with recessive disease? It can be common to have carriers in the population. And the reason I guess I know a lot about this is I am actually this bottom left female. I am a colorblind affected female because my father is colorblind. My mother is a carrier, and I inherited two versions of the gene. I inherited the X chromosome from my father with the colorblind mutation on it, and I inherited it from my mother as well. Both my sisters also inherited the red chromosome from my mother. They could have got the black, but all three of us got the red. And so now I only have two red versions of this gene. So if, I have two sons, so they are both going to be colorblind because their father gave them a Y chromosome and I only have a colorblind X chromosome to give them. So I wanted to give an example in terms of in genetics, how we look at this. So what we see on the right here, this is called a pedigree. And effectively, basically, this is how we as geneticists track inheritance through a family. So it's like a family tree. So males are indicated by squares, females are indicated by circles. And if we see up the top, so we have a male and a female, and they have had two children. And then those children have then had two further children. So this is how we look at families in genetics. So I'm going to go back again to inheritance, the example of the ABO blood group. So we've got those three versions or three alleles, A, B, and O. A is dominant. So we only need to inherit one copy of A to get A blood type. B is also dominant. So if we inherit a copy of B, we will have B blood type. O is recessive, so you need to have inherited O from both parents before you actually have blood type O. And just to make it a little bit more confusing, A and B are actually co-dominant. So if you get one of each, you actually have AB blood type. So if we look at this male, he has blood type A because he has an A version of this gene on his chromosome. The female that he's had children with, she has blood type B. Now their daughter inherited an A from the father and O from the mother. She also has blood type A. However, the son inherited the O's from both parents. So he has recessive blood type O. He has then had children with a female who is A blood type, and both their children will be A blood type because they can only inherit the A from the mother and the O from the father. Okay, so that's genetics. Now, for the last part of what I'm going to talk about is probably what everyone's here for, is actually the genetics of ALS and MND. And I'm going to use ALS and MND interchangeably because in Australia we frequently call it MND. So if I will call one or the other. So first, why is genetics actually important in MND? And we've had, there are many clues as to why genes are important in developing the trait of ALS MND. And one of them's right here. In 10% of cases, in 10% of ALS MND cases globally, we see large families with clear inheritance of disease. So this is autosomal dominant inheritance. There's clear inheritance of MND ALS from generation to generation. So it's obvious that in the hereditary form of ALS MND, there are genes that are underlying the disease. In sporadic disease, however, generally when you think of sporadic, it pops up randomly in the population. But Professor Al-Shalabi, who is speaking next, he did an amazing study a decade ago looking at the prevalence of MND in identical twins versus non-identical twins. And we see in identical twins, the co-twin had ALS MND a lot more often than in non-identical twins, which are effectively siblings. So this in itself was called heritability and shows that there's an underlying genetic basis even to sporadic ALS. Now, I'm actually loath to call it familial and sporadic because there's more of a spectrum of inheritance across this. Historically, we've called it familial and sporadic, and I guess for the purpose of the rest of the talk, that's probably what I will call it, just for ease of use. But it's not really what we see in practice. We have families that don't have a clear mode of inheritance, but they do have more than one person in their family that has ALS or MND. And we also sometimes see apparently sporadic cases that have a gene mutation that we know causes familial MND. So these are clues that there's genetics that underlie, that genetic mutations are underlying the trait of ALS MND. And it's important for us to actually find these genes. So not only is it important, say for diagnosis, if we know what gene and what defect in that gene is causing ALS MND, we can offer a diagnosis, and it has profound genetic counselling implications, which you'll hear about in a fortnight at that webinar. But it also helps us understand the disease biology. So if we know what a gene's normal protein looks like, but we can now look at what the mutated protein, so what this abnormal protein that's caused from a defect in the genetic code. And if we understand what that mutated protein is actually doing, it helps us understand the disease biology, which then can help us lead to treatments. So as geneticists, it's really important for us to find genes. So how do we do it? So this, when we have large families like this, a historical method called linkage analysis, which it's basically we get DNA, so we get blood from everyone in the family that we extract their DNA, and then we look through their entire 3 billion base DNA unit genetic code to find a defect. So we're, say in this example here, with this family, we've maybe narrowed down a region on a chromosome that is shared that is common to all the people that have MND ALS and not in common with everyone else in the family. So we've narrowed it down to maybe one of the volumes. We then have to go and actually look at the full genetic code and try to find the difference. And we can see at this single nucleotide, this single DNA unit here is a T, and in all these people in this family, they actually have a C here. So this is a mutation in a specific gene that's leading to MND. Now, I say this trivially, this takes a very long time. This is effectively what we do in the hereditary form of disease. It's different in sporadic disease, because these individuals have presented at the clinic without any clear family history of disease. So what we're looking for here, we're really doing a statistical test. So we use brute force. We have tens of thousands of MND cases, and we compare them against non-ALS MND cases, controls population, to see if certain versions or certain mutations in the DNA code are more prevalent in ALS MND. And we have, we're part of this amazing global initiative called Project Mine, which is doing exactly this, collating genetic data from tens of thousands of cases worldwide. So this is an amazing initiative to bring all the ALS MND geneticists all around the world together. So now what we've actually discovered, so this figure here, which I have taken from Professor Al-Shalabi's one of his papers, this figure here really shows decades and decades of work of hundreds of genetic researchers all around the world, and in just one small little figure. And so this was in 2017. These are all the genes that we found and the time that we found them. So to date, we've found more than 30 genes that are either associated with disease or actually causing disease. And we generally have four main genes that we talk about. And I'll come back to these in a second. I wanted to show you the prevalence of the mutations in these genes just in our Australian ALS families. So the most common cause are mutations in C9orf72. So we have around 40%, over 100 families that have mutations in this gene. The second most common gene is SOD1 or SOD1. And then we have a handful of families that have mutations in other genes, but we still have a large proportion unknown. So I want to come back to the picture on the left now to look at the timeline of this gene discovery. The first one in 1993 was SOD1. Now, this is the second most common gene. It's not the most common. So that was found in 1993. The next major gene wasn't found until 2008, and Professor Al-Shalabi and we were part of a collaborative Australian-UK research team that found mutations in TARBP. And this was the next major gene and really a changing point in further gene discoveries in ALS. We found FUS as a collaborative team 12 months later. And then 2 years after that, we found the defect C9orf72. So researchers in the US found the defect in C9orf72. And it's strange because this is the most common genetic cause, but wasn't found first. And the reason is, you might remember back, I said that strange repeat expansion. That is what is the defect in C9orf72. And it's from a molecular biologist perspective, it's a strange mutation and difficult to find, but it is the most common cause of both familial and sporadic ALS. So can both familial and sporadic ALS cases have gene mutations? Yes, absolutely. I will give you an example. So the SOD1 gene, this is the DNA code for the gene. So it's 465 DNA units long. So this is the actual genetic code for SOD1. And these are all the mutations that have been found in there. So along the bottom where there's a rectangle, that's representative of the gene itself from the first DNA unit to the 465th. And these are all the mutations that have been found in ALS and MND familial cases in orange and sporadic cases in blue. So this is the second most common gene. If we now look at C9orf72, which is the most common, there's only one. So that is this repeat expansion in C9orf72. And there are hundreds and hundreds of MND cases and families all around the world with this specific mutation that's come from a common founder hundreds of years ago. So that's, so yeah. So to date, how many ALS or MND linked genetic variants have been reported? So these could be mutations or just variants that are associated with disease, so may not cause disease. And more than 1,000 have been reported. So we have some really amazing online resources to actually, for anyone to actually go and have a look at the genes and all of the variants that have been reported in ALS and the research papers that have first reported those genes. So ALSOD, which was set up by Professor Al-Shalabi's, some of his research team in the UK. So they've put this database together. There's also a new NIH-funded initiative called ClinGen, which so ALS has recently been added to it this year, and the purpose of this website is to obtain, this initiative is to obtain evidence from medical research on defining the clinical relevance of all of these genes. So whether they're actually definitive ALS genes, or maybe the evidence is limited, that they may be associated with a disease, but not definitely causing ALS. So this website is available. To date, 10 genes have been done. And lastly, I wanted to give a few examples of actual inheritance, modes of inheritance and inheritance patterns in ALS MND. So autosomal dominant inheritance, which as a reminder, you need one copy, so one version of your gene that has the defective mutation to get disease. That's autosomal dominant inheritance. The majority of ALS MND genes in families are autosomal dominant. So here's an example. These are 3 Australian families that have a mutation in FUS. And we can see from generation to generation, there is inheritance of MND. So there's actually been one gene reported on the X chromosome, and that's called ubiquitin 2. And again, this is an example of an Australian and New Zealand family that has a mutation, a disease-causing mutation in ubiquitin 2. And so this looks very similar to the pedigrees on the previous page, where we're seeing dominant inheritance. But the reason that we know, there's 2 reasons we know it's excellent. Is 1, ubiquitin 2 is on the X chromosome, but 2, looking at this pedigree, you never see the MND passed from father to son ever, because that does not happen in an X-linked gene. So everywhere where a male is affected, it is never passed on to his male sons. And lastly, we have autosomal recessive. So this, the gene alsin 2 it causes juvenile ALS with an autosomal recessive inheritance pattern. So juvenile is when ALS, typically symptom onset is before the age of 25. So we have these families. So this was reported long before I started working on ALS. And we see in the bottom generation individuals that are coloured in black, which means that they have MND, and their parents have half black, half white. So they carry a mutation in alsin 2 that is causing disease. And an autosomal recessive pedigree wouldn't generally look like this. There are a lot of people affected in this family, but they're only in the one generation. And the reason that we're seeing a lot of people in this family affected is these double lines refer to a consanguineous marriage. So these people, these parents that are carriers are actually related. So they may be first cousins, second cousins, or even or higher than that, closer than first cousins. So that, I guess, wraps up the basic genetics, of basic genetics and the basic genetics of ALS MND. So I'd like to now pass over to my colleague, Professor Ammar Al-Shalabi. So thank you.
Thank you very much for that, Kelly. It was very, very difficult to summarize such a complex topic so well. So thank you for doing that. It makes my job easier. I will continue the theme of trying to answer questions using our template. And like Kelly, I will keep the complex words highlighted. And the reason we're using the complex words, the jargon, if you like, is because you may well come across the jargon yourselves when you're receiving, for example, information about genetics. So a common question we're asked is that the person carries an ALS gene mutation. Does that mean they will definitely develop ALS? And that's an issue which in genetic terms is called penetrance. It's how penetrant the gene mutation is. And in simple terms, penetrance is the risk that you will develop ALS if you carry an ALS gene variation. Most ALS gene mutations show what's called age-dependent incomplete penetrance. So what that means is the risk of ALS will increase as the person ages, but not everybody who carries the gene variation that would normally cause ALS will necessarily develop ALS. So what you can see on the right here is a pedigree from my colleague Peter Anderson. It's the same as the pedigree style you saw from Kelly, but it's been made into a circle because it's such a large pedigree. And I want you to just look at the people that are highlighted in red here. If you look at that part of the pedigree, so it's slightly upside down, it's at an upside down 45-degree angle, so I hope you can turn your heads or visualize it. But the father, who is in black at the top of the pedigree, has had a son who's then had more children, one of whom is affected, and that person has also had children, one of whom is affected. And so if you were the person at the bottom of that pedigree and you came to clinic, then you would say, yes, my father had ALS and so did my great-grandfather. And so it would seem like it was familial ALS. Now in the same pedigree, and we know all of these people have the same underlying genetic cause, you have two people here who are very distantly related from their point of view. I think they're sixth cousins. So it looks like sporadic ALS from their point of view. It's very unlikely they know of each other. And above them are at least 6 people who carry the disease gene variant, but do not develop ALS. So that shows you that even within the same pedigree, the degree of gene penetrance can vary. And part of that will depend on how long each of the parents live for, but it also depends on other factors that we don't fully understand. So that means that genetic ALS isn't inevitable. If you carry a disease gene, it doesn't necessarily mean you're going to develop ALS. And the reason for that is that we know that 6 molecular steps are needed to develop ALS. And mutations can account for up to 4 of those steps. They don't necessarily count for 4, so some of them will account for just 2 of the steps or 3. But even if they accounted for all 4 steps, you still need 2 more steps to happen. And those are caused probably by lifestyle or environment or aging or something that we don't fully understand. So you can carry the disease gene that greatly increases your risk because it's taken your body through 4 of the 6 steps that are needed to develop ALS. But something else still needs to happen to develop the last 2 steps for you to then develop ALS. So you can carry an ALS gene and never develop ALS, but generally, people who carry a gene variant that is strongly linked with ALS have a much greater risk of ALS as a result. Now we know that ALS varies between people. It can affect different sites of first symptoms. So somebody might develop speaking or swallowing problems first, or a problem with manipulation of small objects, or lifting their arms, or a dropped foot. It can affect people at different ages. So the youngest person I've seen was 16 and the oldest was more than 100. The progression rate can vary dramatically. So some people can have extremely aggressive disease and become severely disabled within a few months. And other people can have extremely slow disease progression with very little change over many years. So there's a lot of variation just in those kinds of clinical pictures, but also the degree of neurological change, which we call upper or lower motor neuron involvement, which is really telling you about how stiff the muscles are versus how wasted they are. And that's to do with the particular nerve groups that are affected. Even that can differ between people. And in some people, there is also impact on thinking or behavior, so cognitive change. And we know that some mutations in ALS genes can directly influence that ALS pattern. But we also know that variations in other genes that don't necessarily seem to have any direct effect in ALS can also modify it. So one example of a direct effect is that nearly every big gene variation, so by that I mean a gene that has a big effect. So if you carry that disease gene variation, you're very likely, but not definitely, going to develop ALS. So if you carry one of those disease gene variations, then your age of onset is likely to be lower than average. And what you can see on the right there is the probability of a positive gene test by age. And you can see that the younger you are, the more likely you are to have a positive gene test. That's just reflecting the fact that the risk genes lower your age of onset. You can also have specific changes in the clinical picture that are more likely depending on the gene variation you have. So Kelly mentioned this important gene C9orf72. People who have ALS caused by that gene, by variation in that gene, are more likely to have speaking or swallowing problems as their first symptoms and as a predominant feature of their symptoms. And they're also more likely to have cognition and behavior affected, but neither of those is inevitable. It's just a tendency. People with mutation in the SOD1 gene, which affects about 2% of everybody with ALS, are more likely to have a lower motor neuron pattern of their disease. So that means they're more likely to have wasting or floppiness rather than a stiffness to the pattern of ALS. And within people with SOD1 mutation, the actual variation the person has inherited can also directly affect the clinical picture. So for example, somebody who has inherited a disease variation called A5V, it used to be called A4V, people with the A5V variant of SOD1 are much more likely to have an aggressive course of disease, whereas people with the D91A version of SOD1, which used to be called D90A, are more likely to have a slow version of the disease. Now, these numbers and letters, A5V or D91A, they're referring, the first letter is a code for the amino acid that should be at the base, at that position in the protein, and the second is the amino acid it's been changed to. So the building blocks of the protein, the actual protein sequence is changed, and this is just a code to make it clear how that change has happened. FUS, which was one of the other big 4 genes that Kelly mentioned, is much more likely to cause ALS at a young age and is also more likely to affect thinking. So the reason there's a picture of a waterfall, it's actually the Iguazu Falls on the right, is to show you the kind of, to give you an analogy of what this means. So if a river is along its course and there's a break in that course in height, then you'll have a waterfall. So if you like, that's like ALS. And then some waterfalls will be short and other waterfalls will be very high. And so that's an example of a direct effect of the thing that's caught, the actual thing, the waterfall, the size of the waterfall it's a direct effect, a direct relationship between the two. But you can also have genes elsewhere in the genome that modify whether how ALS behaves. So for example, a gene called UNC13A, we know that people who carry variation in that gene have a small increase in their risk of ALS, but also if they develop ALS, they're much more likely to have a rapid disease course. Variation in the CAMTA1 gene doesn't seem to influence your risk of ALS at all. But if you happen to have ALS and you happen to have a particular variant in that gene, then that will affect your rate of progression. It can either slow down ALS or speed it up. So that's a bit more like whether there's been a monsoon or a drought or the season. It's an indirect effect on the waterfall. It's the same kind of thing with these genes. They're influencing the way that the main ALS gene is manifesting. And these genes actually seem to have an effect even if you don't have an obvious genetic cause for your ALS. So they influence ALS in general. ALS genes can also cause other conditions outside ALS. And when we have a gene with multiple effects, we call that effect pleiotropy. So you may see that sometimes in reports or in things that you read. So genetic pleiotropy, examples of that in ALS are the 3 genes listed there, C9orf72, FUS, and TARD-BP. They all are also able to cause frontotemporal dementia without ALS. And the ataxin 2 gene, you have a repeat expansion in that. Kelly showed you a good example of repeat expansion earlier. So if you have a repeat expansion in ataxin 2 that's very large, then you develop ataxia. But if you have one that's intermediate in size, so it's not the more normal population size, and it's not the very large ataxia size, but somewhere in between, that increases your risk of ALS. So these are genes that have multiple effects. And we know that genes contribute to ALS risk in general. As Kelly said, the heritability is about 60%. So a way of thinking about that is that you have many small effect gene variations, each one nudging you towards developing ALS. I've got a picture of the Milky Way there because it's, although it looks like a single object, it's actually thousands or millions of stars, each one contributing a little bit of light and therefore making a milky appearance. It's the same kind of thing. It's lots of small effects of these gene variations because we all have thousands of them. And depending on the particular combination you've got, that will increase or decrease your risk of ALS on average. But more usually, we look at ALS with these large effect gene variations that are illustrated in the pedigree here. So this is a real pedigree that was one of the pedigrees that led us to identify the C9orf72 gene in people with ALS. So people with sporadic ALS can still carry familial ALS gene variants, as you heard from Kelly. And that's because statistics means that smaller families have less chance of a positive family history. So the two branches of this family that I've arrowed, there aren't many people in the offspring group in the lower parts of the pedigree. And so there's just less chance of either inheriting a risk gene, or if it's inherited, if it doesn't have 100% penetrance, again, that reduces the chance that ALS is going to manifest. And so having smaller pedigrees, which is now the trend across the world, people are generally having smaller families. It just means that there's less of a chance of a positive family history. Pleiotropy also plays its part in meaning that people with apparently sporadic ALS can actually carry familial ALS gene variations. And that's because, for example, if you look at the arrowed part of the pedigree here, the person in blue who has ALS has a parent with frontotemporal dementia rather than ALS. And so if you weren't aware of the link between frontotemporal dementia genetics and ALS genetics, then you might say, well, that's not a relevant family history. This is just sporadic ALS. And that was certainly what we used to do until the 1990s. It was not really regarded as relevant if somebody had frontotemporal dementia in the family history. And there may well be other conditions now that we don't regard as important in the family history that are actually important because they have a common underlying genetic cause. And finally, reduced penetrance. So the circled part of the pedigree there, gene carriers don't always develop ALS. So there's a parent there who must carry the disease gene variation, but it hasn't been affected. So if people with apparently sporadic ALS can carry a disease gene or have an increased risk because they carry lots of small gene variations all nudging towards developing ALS, then does that mean there's an increased risk to relatives, even in people with sporadic ALS? That's a really important question. It's one I'm asked a lot in clinic. So we can answer it like this. So the lifetime risk of ALS is about 1 in 300. You can see on the right, there's a graph. So on the x-axis is age that's increasing, and on the right is the percentage risk of ALS. And you can see the red line is the 1 in 300 risk, so that by the age of about 90, there's a 1 in 300 risk for men, and it keeps going up, becomes about 1 in 220 by the time you reach 100. And for women, it also, the black line, keeps increasing, not quite as fast a rate. But it's difficult to be sure at the very old ages because we have fewer people coming to clinic at those old ages, either because there have been competing causes of death or simply because very old people, often the primary care physician will say, well, I don't want to trouble them with going to a clinic. And so we don't see those referrals. Now, we then asked whether relatives of people with apparently sporadic disease have an increased risk. And the answer is there is a technical increase in risk, but in practice, it's almost negligible. And people who have a relative with sporadic ALS, apparently sporadic ALS, still have an overwhelmingly likely chance of dying of heart disease, stroke, or cancer rather than ALS. And you can see that on this graph. So this was a study we did where we actually went through our clinical notes and we saw where people had come with apparently sporadic disease, later on was that diagnosis changed to apparently familial. And the gray line is the background risk. So that's the 1 in 300 line. You can see it does decline. That's just the risk to the population. And the black line is the risk of ALS in a first degree relative of somebody with apparently sporadic ALS. And you can see it does go down, but actually, if you look at the y-axis, it's very, very, I've zoomed into the very, very top part of this graph. So even at the bottom, it's 0.95. If you go to the bottom of the screen, it's about 0.91. this graph, if you did it at the scale, if you're looking at a normal computer screen or TV screen, it would go down at least 2 meters to the bottom. So we've really zoomed into the very top. Those two, the black and the gray line, are actually very, very close together. They're almost impossible to distinguish. So that tells you they do have an increased risk technically. And a good way of thinking about that is if I buy 8 lottery tickets, I've increased my chance of winning the lottery jackpot by 8 times, but I'm still not going to win. So it's a bit like that with this. You increase your risk of developing ALS a few times if you have a first-degree relative with apparently sporadic disease, but the risk to start with is so small that it doesn't actually make any difference in practice. So now we know that ALS can be genetic or not genetic. What can we actually do about it? Well, one option is to target people with specific genetic subtypes of ALS, and that's called precision medicine. One example is of lithium in people carrying the variation in that gene I told you, UNC13A, that slightly increases your risk, but actually if you carry that variation, the big effect of that gene variation is that it gives you more aggressive disease. The graph on the right is a survival curve. So if a line is more horizontal or more to the right, that represents better survival, and a line that's more vertical or coming down towards the bottom of the graph is representing worse survival. And what you can see is this pinky purple line, depending on the colors of your screen, is low, and the other lines are much higher. So the other lines, the other 3 lines, represent the normal course of ALS, and the purpley line represents people with UNC13A variation. And in fact, that purpley line, this is taken retrospectively from 3 different clinical trials from colleagues in the Netherlands, Italy, and from our study in the UK. We were able to show that people given lithium actually improved their survival from this pink line back to the same survival as everybody else, whereas people who were given a placebo maintained their poor survival. But when lithium was analyzed, when these clinical trials were analyzed generally, the question wasn't anything about genetics. The question was, does lithium improve survival in ALS as a whole? And the answer is it doesn't. So it's interesting that within that subgroup, within that clinical trial, you're able to pull out a subgroup of people with a genetic basis for their more aggressive disease, and the lithium does actually seem to treat that subgroup of people. So we've now got a phase 3 trial looking at whether lithium therapy in people carrying this genetic variation might be a good strategy. You'll have probably also heard about gene therapy. So there's lots of ways you can target treating people with a genetic basis for their ALS. One method is called antisense therapy, or ASO therapy. ASO stands for antisense oligonucleotide, and an oligonucleotide is just a short stretch of RNA or DNA. Now, if you can deliver this antisense piece of genetic message into a cell, then it can block a gene from working. So what you do is you make a piece of oligonucleotide, a piece of DNA or RNA that exactly matches, it pairs up with the disease gene variation that you want. And by pairing up with it, it signals to the body that it needs to be destroyed. And then the body destroys that genetic variation that would otherwise have caused disease. Antisense therapy is now in phase 3 trial. At least one phase 3 trial has just completed. We're still waiting to hear the results from it, but the phase 2 trial looks very promising based on some biomarker studies. So we're optimistic that this might be a really effective strategy for treating people with ALS. Antisense oligonucleotides do require repeated treatments and lumbar puncture delivery. So it's not like taking a tablet, but it is at least a potential step forward that would be really remarkable if it works. There are other ways you could target genes. So you could make antibodies that, for example, target only mutant SOD1 protein. And then if you can deliver those to the cells, then they will destroy the mutant SOD1 protein. You can also use small molecules. So that would be an example. Lithium might be working like this, where the small molecule of lithium goes into the cell and then somehow affects the gene variation or something to do with the way that gene is working. And then other strategies, one that's being looked at is packaging genetic signals, genetic strategy, genetic treatments into a virus. And AAV9 virus, adeno-associated virus 9, doesn't normally cause disease in humans, but it is motor neuron specific, and so it's been used as a way of doing this. So you can package the gene sequence you want inside the virus, and you can deliver it, and then the virus delivers your payload into the motor neurons, and then it can do its gene therapy there. So the advantage of that is it's one treatment only, but you could say that's actually a disadvantage if you don't know about long-term side effects. So just to conclude, genes, as Kelly showed you, carry the instructions to make our bodies, and they are inherited in all the different ways that she showed you. Changes in the gene sequence can lead to ALS, and those genes can affect risk even in people without a family history. And we can use genetics to guide treatment. Thank you very much for listening.
Thank you very much, both of you, for such fantastic presentations, explaining some difficult concepts to grasp. And I think both of you explained everything in such a clear manner. I hope that this is a valuable resource for people living with ALS and their families. We've had a couple of questions that have come through, and I'm also going to put the link to the feedback survey in the chat for everyone. The first question that we had that came through was the pedigrees that you used in your presentations. Do those represent actual families with MND ALS, or are they just illustrative purposes?
Kelly, you're muted, so if you want to go first, yeah.
Yes, so the families, the pedigrees that I had used, all of them bar one were actual Australian families with ALS and MND, so yes. And
the same for the pedigrees I used. Wherever I gave a reference, those were from real families, but they were anonymized in some way, so the male-female structure was changed, or the number of children modified, so that it makes them more difficult to identify.
Yeah, so our Australian ones had all been pre-published.
Yeah.
Perfect, thank you. We also got a question about what we mentioned earlier about some resources being developed and translated into multiple languages. I'll take that one. And so the Alliance will be working with its member associations to start with the languages that serve those clients first. And so that's how they'll be proceeding with the development of resources. Bruce, did you have a question that came through?
Yes, I do. The question was, is it possible to have multiple ALS genes? And also, how do these mutations cause selective loss of motor neurons? The second is probably a difficult one. Over to you.
Who would you like to answer that, Bruce?
I think with the wealth of expertise, perhaps Emma, if you could answer the one about possible to have multiple ALS mutations, and Kelly, perhaps you could answer the one about how those mutations can cause selective loss to the motor neuron.
So the simple answer is that you can carry multiple ALS gene variations that each increase your risk. You can carry, for example, you might carry the variation in C9orf72 that increases the risk of ALS, and have a mutation in another ALS gene like SOD1. That does happen. It's rare. In most studies, it's about 1 or 2% of people seem to carry multiple variations in ALS risk genes.
Yep. I'm wondering if I can actually add to that a bit, because we did do a study, we did an Australian specific study on that exactly last year, and as Amar said, we didn't see any that had any sporadic MND cases with multiple major ALS genes, but we did see individuals that would have, say, a major ALS gene like C9orf72, and even up to 2 other gene mutations that had been shown that have been associated with disease, including ataxin-2 intermediate expansions. In terms of the other question, I actually can't answer that. I'm sorry. I don't know. I don't know why they are causing selective loss of motor neurons. And I don't know. Perhaps your research team may know some more exciting things about that. But I think if we do know that, it's wonderful news for developing treatments.
Yeah, so we've got a clue as to why it might be, but it's never been proven. And that is in the multi-step model of ALS, which is this model I told you about, where we've shown that 6 steps are required to develop ALS. It's quite likely that at least one of those 6 steps can only happen in motor neurons because of the specific things that make a motor neuron. So we can't be any more specific than that. But for example, if one of the steps requires the presence of an androgen receptor on the outside of the cell, we know that those are seen on motor neurons. They're not really seen on many other nerves, on many other cell types in the same way. So for example, that might be the kind of thing that's required to make that molecular step happen.
Thank you.
Thank
It
you.
was a difficult question and possibly beyond the scope of today's introduction as well. Thank you for both.
We have, I think, one more question that has come in. If a patient has ALS, is it usually caused by a mutation in a gene? I think they're asking, are the majority of cases of ALS caused by a mutation, or is it not the majority? I believe that's how I'm going to interpret that question.
Kelly, do you want to answer that one?
Kelly?
Oh, I was going to pass it on to you, but
Okay, I'm happy to do it if you want.
yes, you deal with patients. You
Okay, so about 5 to 10% of people, as Kelly showed in her talk, have a strong family history of ALS, and in most of those people, about 80%, we can find a big ALS gene, a gene that has a big effect and greatly increases your risk of ALS. For the 90 to 95% of people where we don't have a positive family history when they come to clinic, that doesn't mean that they don't have a genetic cause, but probably about 10 to 15% of those people have one of those familial ALS genes, one of those big effect genes underlying their ALS. For the other roughly 80% of people that are now left, they have probably a genetic contribution, but it's more like that Milky Way thing I showed you, where there's lots of small genes that are all nudging them towards developing ALS, but none of them is big enough to actually cause ALS on its own. They each just slightly increase the risk. So the answer is probably about 15 to 20% of people in total, of everybody who comes to clinic, regardless of family history, has some strong genetic basis for their ALS. And for the remainder, there is a genetic contribution, but it's not the main driver of their ALS as far as we can see.
Thank you. So we have no more questions that have come in, but I have one final question to kind of bounce off of that strain and to close us up today. What you presented, Professor Al-Shalabi, on the UNC13A, precision medicine, looking at a genetic alteration that could, related to survival, and targeting that specifically with a therapy. Given the number of clinical trials that are currently ongoing, not specifically for the genetic forms of ALS, do you think that's a strategy that we're going to start to employ more and more?
It's very difficult because immediately you end up with trials competing with each other. And it's a nice place to be in for ALS that we have enough trials that we can do that, but it does also come with its own problems. I think it is a strategy we'll see more and more as these genetic subtypes open themselves up for specific targeted treatment. The issue will be then, can we also use some of those gene therapies in people who don't have an obvious genetic cause? Because they may have the same pathway affected, even if they don't have the same cause. It may be the same problems in the cell, and if you target the same gene therapy, that might work even if you don't have that genetic problem underlying your ALS. We don't know. So it may be that in the end, these types of gene-specific therapies or targeted genetic therapies are actually used much more widely.
So really, genetics is the answer to everything.
No, genetics is the answer to a lot. It's an important step forward.
Well, thank you so much again. I'm just going to share the feedback survey for everyone in case they haven't had a chance to copy it from the chat. Thank you both so much for joining us today, for providing these presentations to the community, and they'll be recorded. They were recorded and they'll be on the Alliance's website within the next couple of days for anyone who missed a portion or wasn't able to attend. We're very grateful that you were able to join us today. Again, thank you so much.
Thank you.
Indeed. Thank you both. And thank you, Christiana.
Thank you, Bruce. We'll do it again in 2 weeks. Thank you, everyone.